Yo, okay, are we live? I think we're live. If anybody is here in the chat, please let me know if you can hear me. If you can't hear me, then that's something that we should figure out soon. But I'm assuming that you guys can, so, yeah, let me just put that in the chat just in case. Can you guys hear me? Again, so this is my very first live stream. I'm very excited to be here, and I'm very excited for you guys all to be here. Some of you might be new to the channel, and some of you might be long-time subscribers. Either way, you are very welcome to be here. This space is for people that are new to science completely, and as well as some experienced microscopists as well. So, yeah, so what I'm going to do is I'm just going to kind of wait for a couple of minutes for people to trickle in, and as that happens, there's just one more thing that I'm going to test out. I wanted to see if I could add background music to this, and so I'm seeing in the chat, looks like you guys can hear. That's awesome. Good. Let me know if you can hear this background music. I'm going to turn this up a little. Or have you been hearing background music this whole time? Yeah, so here's some music. I'm just hitting play. And let me know if it's too loud or too quiet. Just gonna let that play out for a little bit. And then in the meanwhile, I'm going to be zooming in here on something that I wanted to show you all. So again, let me know if you hear some music. If you don't, that's totally fine. We can do it without music today. Again, this is just very new to me. Never done live streams. No music. Oh, that's fine. Yeah, let me try one more thing. Yeah, it's weird. It says it's active, but cool. What if I like turn this way up like this? Do you hear music coming from there? Anything? Bueller? No. Oh, my mom says she's been hearing it. I mean, random audience member. Okay, this is music at half of the volume. Is this music good enough? Like, I don't want it to overpower me, just like whenever I'm speaking, but I do want it to be able to like fill the void and stuff like that. Just let me know. And then, okay, cool. Yeah, and then just let me know if it gets too loud or too quiet. Okay, let me turn it up a little bit more. Okay, I'll just keep referring back to the chat, and if you guys say like, oh, it's too loud or too quiet, I'll adjust the music and we'll figure it out. One of the other things too is that I'm going to be switching into a couple of different scenes and stuff on this live stream, and the music will only work on this main show thing that we're doing. So it'll turn off in just a second, but this is kind of like the main experience that people will have for most of the show. But anyway, it's been about 5 minutes. Oh, random audience members says good right here with music. Okay, perfect. Awesome. So, yeah, I think that's good. Music, we've got lighting, we've got microbes. My son is asleep. Hopefully he stays asleep throughout all of this, and if not, we'll have to do a little intermission, but that's totally fine. So, yeah, so before, oh, okay, now they say it's too loud. Okay, let's turn it back to this. Okay, again, lots of trial and error with this, but we're going to figure it out. And, yeah, okay. So it doesn't have to be, it just has to be like there. If it's but yeah, we don't want it to be overbearing. So yeah, okay. I'll just keep it right there for now. And let's talk about some microbes. I mean, we're already 5 minutes into this thing, and I mean, who knows how long it's going to go, but so you guys are welcome to stay for the whole thing. For any of those people that I've asked siblings and family to help with just checking the audio and visual, you guys are free to go if you want, but I really would love the support if you guys want to hang in there. But yeah, so before I go into the spiel here, I wanted to share a little story, a little microscope story. So right now, you guys are looking at some microbes under the microscope, and let me zoom out just to show you. I'll zoom out and then adjust the focus here. These guys are really small. You can barely see them with the lowest zoom. You go in a little bit higher, and here you can see them a little bit more clearly. And now we're going to go into my highest zoom available. And yeah, so this is the closest up I can get with these microorganisms. So these guys are called Dunaliella. I think that's what they are. Let me just double check. Yeah, Dunaliella. Dunaliella. So these microbes are actually what, so there's a couple of things to know about them. One, they are unicellular algae, so they're kind of more closely related to plants. Two, they live in a very, very extreme environment. And for any locals that are watching the show, I'm based out of Utah. These guys are found in the Great Salt Lake. And the interesting thing is that you can actually, even though with this microscope, you can barely see them, you know, when you're zoomed out, I'm telling you right now, you guys can see these guys from space, if there's like a large enough group of them. And the reason that is because of a couple of things. One, the Great Salt Lake is very, very salty. There's not a lot of things that can survive in there. And so there's not a lot of competition when it comes to surviving. So there's not a lot of other algae to outcompete for sunlight. And then the only thing that eats these are brine shrimp, brine flies, and birds here and there. But there's not a lot of fish. In fact, I don't think there's any fish in the Great Salt Lake. It's really just the brine shrimp. So, oh good, you guys are naming them, that's great. Yes, they're very fun to watch. They have tiny little red spots on them that can sense light, and those red spots, they can't detect shapes or anything, but it does help them go towards light, and so they can photosynthesize and survive better. Anyway, so the other part of this story is that this is, there's like a sister species to this. They're both called Dunaliella. One is called Dunaliella viridis, and then the other one is called Dunaliella salina, or something like that. Somebody can fact check me, or maybe the auto captions will work. But let me just take a brief pause here. I'm going to pause the music, and I'm going to switch over to a PowerPoint to show you guys something really cool. So, and I'll go back to those guys in just a second, but I wanted to show you this. So this is the Great Salt Lake in Utah. It's the largest lake west of the Mississippi, besides, you know, Great Salt Lakes, or the Great Lakes. I don't know if any of the Great Lakes are west of the Mississippi. Anyway, it's a big, big lake. And one of the first things that you'll notice is that there is clearly a division among, in color in this lake, which is kind of interesting. Are you guys seeing this PowerPoint, by the way? I'm testing out switching screens, so I think this is working, but just wanted to double check. Yeah, I think it's salina. And And then the other one is called viridis. Sorry, just answering some things in the chat there. Anyway, so, okay, cool. So PowerPoint's working. Yeah, so here's the Great Salt Lake. On the bottom, this is where I collected that green algae. So these guys are living in about 12% salt water. Now, for context, I think the ocean is like 4 times less the amount of salt water, or like has like 4 times less the amount of salt in the water. Now, the reason why the top is different is the top is the different species called Dunaliella salina. And yeah, so to John's point, it is different mineralizations, but the other thing is that due to the snow runoff, the bottom half gets a lot more fresh water than the top half. And this used to be just one single colored lake, but what happened was, if you look at the top right picture here, this, as you can see, oh, sorry. In the top right image, there's a big train track that was built, I think like in the 50s or 60s, that went straight through the lake. And once that happened, the freshwater runoff wasn't able to get into the lake as much. Now, it still gets some runoff here and there, but what happens is that the top half ends up being a lot saltier. I think the top half might be around 18% salt water. Again, these are numbers I looked at a couple weeks ago, but I don't remember off the top of my head. So anyway, just kind of a cool thing. Like, you can see microbes from space. Check this out on Google Maps, on Apple Maps and stuff. You might be able to see it. It doesn't work at every single satellite image, and I think the reason is that they might have been taken at different times of year, and so it's not always like this. But yeah, so there is a very cool little intro to something really cool about microscopy and about microscopes. So this is actually part of a episode that I'm going to be releasing soon, and it might be part of a series because last week I just went and collected some microbes from the south side, and then eventually I got to get to the top. But it's a bit of a journey. I mean, just to get to the south end of the lake, it took me about an hour and 40 minutes just from my house. And so just to get all the way to the top to get those microbes and come back, that's going to be a bit of a journey. It'll be like 5 hours, 5 or 6 hours. Are you guys still hearing me all right? Am I too loud? I can turn myself down a little bit. Cool. Seems like you guys are enjoying this, maybe or not. I don't know. So let's move on. We're about 10-15 minutes in, and so hi Matt D. Welcome to the show. And Matthew, yes, looks like an algal bloom. It definitely could be at these certain times. Algae blooms are extremely common in Utah because we get lots of sunshine and we have lots of shallow lakes. So we've got the Great Salt Lake, which is really shallow almost all the way through, and then we also have Utah Lake, which Utah Lake is only about 8 or 10 feet deep on average the whole way through. So it gets a lot of sunshine, and the algae do kind of take over very easily. Yeah, make sure you guys have those notifications on for the stream. So, okay, let's keep moving on because I have a lot of really other cool things to show you guys. And so I want to move on with this PowerPoint here. I've just got a couple of slides as kind of like an introduction. So been about 15 minutes. Glad everyone's here. Welcome to the Microscope live stream. If you were here for the story, about the microbes that you can see in space, that's great. If not, then you guys can always watch this afterwards. This whole stream will be available on my channel immediately after I publish. So this is my very first live stream, and if it does work out, then I might start doing this on the regular every Tuesday night, like at 8, either from like 8 to 9 or 8 to 10. It just depends on how many cool things that we can find. So let's go to the next slide here. Updates. So these are the really big updates which kind of prompted me to want to do this. So I've just crossed the threshold of creating 50 videos, and it's been a lot of work. And so I just wanted to pat myself on the back right there, and wanted to also thank you guys so much for your support, because even like with videos that get, you know, a couple dozen views or a couple hundred views here and there, like it's still important to me that somebody else gets a little bit of joy and a little bit of knowledge from this microscopic journey. And so again, big shout out to you guys, big thank you. Definitely could not have done it without you guys. Okay, just checking the comments again. Looks like you guys are enjoying the volume. That's good. Okay, next thing here. So this is what you can expect in this stream. So I've got a couple of things. I wanted to keep it interesting, you know, start off with a little story there, and then do a little bit of intro about me and how I got involved into microscopy. But I'll kind of be discussing that a little bit as we go into the pond water, and all of this is kind of going to overlap a little bit. But basically, this might be kind of the way that we go about doing these streams. I think we might start off with just some prepared slides to kind of calibrate my microscope and my camera and make sure everything works. And then we'll switch over to pond water and do some live stuff. So, or we might do it the opposite way. I don't know. It depends what people want. So comment on your favorite parts of this, and we can do that. The thing about the prepared slides is that a couple of years ago, I bought this thing from Amscope. This is a collection of 100 prepared slides. And this is normally, you know, they sell this for like science channels and stuff, or science teachers and things like that. But anyway, so it's really nicely prepared. It has, sorry, I'm like seeing myself on the screen and it's bugging me. I gotta hide myself real quick. So it's got 100 slides in here, and they're from all sorts of stuff. So we have like animal parts, like mouse kidney, or like part of a rabbit brain, and then we have like connective tissue and things like that, and then we have like insect pieces, and anyway, I'll eventually put up like a list of like all these things, and then you guys can let me know like, hey, which ones do we want to see in this stream? I actually have 2 of these, and they're both totally different. So we have 200 prepared slides that we can go through, and I was thinking, you know, like for the first like 5-10 minutes of the stream while people are circling in, we could kind of go through these. And so I just kind of want to give like an example of what that would look like. And again, I'll kind of, you know, post, you know, what oh yeah, the motor neuron. I think that'd be really cool. So yeah, let me see if I can find that one here. You know, shoot, I think that one is in my other one downstairs. So, but I'll have a list of which ones I have, and then you guys can choose or like place your vote. But for today's episode, I just have 3 that I have just kind of pulled out and ready to go. And these 3 I've felt like would be kind of like a good introduction to microscopy. So let's switch on over to this, and if you guys want to make your last goodbyes to your Dunaliella, you can say your goodbyes. Oh, there was one more cool thing I wanted to show you guys on this real quick, before we go to those prepared slides. So while I was pulling these up, I found a small little larva of a brine fly. Now, it's small in real life. It's like 1 millimeter long, but let's see. Dude, where'd you go? I swear he was on here. It might just take a second. Dude. Maybe it escaped to the edge. Oh yeah, so this is salt water, so you can see the salt is starting to crystallize on the edges as the water evaporates. Water evaporates pretty quickly on these things, so you only have really few minutes, especially if the lamp underneath generates heat. Man, I swore I had it. Okay. Oh, let me get that music going again to make things a little more spicy and interesting. Yeah, don't drink the water. I'm thinking that might be a good catchphrase. Don't drink the water. Dude, are you in here? I swear it was just on here. Maybe it like dissolved and died away because of the salt concentration or something. Yeah, shoot. Sorry, I'm just kind of zooming a little bit quickly here because I want to get to some exciting things. I'll give it 10 more seconds and then we'll go. 10, 9, 8, 7, 6. Oh, well, that's a bummer. Well, maybe you turned into a real fly and flew away already. I don't know. Do you guys see anything? This, like, it looks like there's nothing on this slide as is. So, yeah, the salt is interesting. You know, I've done a couple episodes of like looking at crystals with like polarized light. You can see I did one on Epsom salt recently. It's Epsom salt under the microscope, polarized, and it's like a total art piece. It's really cool. Ooh, saltiest microbe. That is a good idea. I like that. It would probably be something in the Dead Sea, because I think the Dead Sea is a little bit saltier than the Great Salt Lake. Anyway, oh, sorry. Let me introduce to you this nylon fiber. This is just to show you guys the scale of the microscope for those who haven't been watching my channel and see what is, see what there is. A question. What's my favorite microbe? Probably, that's a tough one. Right now, it would probably be Gastrotrix. They're really small and really, they look kind of fuzzy. Here, let me put like a white thing behind this so you can kind of see. So this is a nylon fiber. The WM stands for wet mount, and you can kind of see with my finger just how small that fiber is. So the cool thing about nylon is that it is a strong plastic, I believe, and so because of the way that it's made, it's able to be woven in really, really small strands really easily. And so yeah, let's zoom in here a little bit more. You can see some really fine detail of this. I don't know if you guys have ever seen how it's made. I think there's a how it's made episode on nylon or clothing, and they talk about nylon fibers and stuff. Really, really interesting. And then this is my highest magnification that I've got, but the problem that happens when I get with this high of a magnification is that there is very little area that you can focus with. And so just even moving up like a fraction of a millimeter, you lose focus. And so doing something that's kind of three-dimensional like this is a little bit tricky to do. But that microbe that we were looking at earlier, that was at the very highest magnification, but since it's so small, it's easier to focus on that one. Another cool thing I can do with this, so this is my bright field lens, and I can adjust the exposure to give you a different view, and I've also got this blue filter on it that can also be used to shine light on it a little bit sideways as kind of like a DIY DIC microscope. There are some really expensive microscopes out there that cost an arm and a leg and $10,000 that make microbes and specimens look kind of 3D, but you can kind of make a makeshift version just by bending the light a little bit here. Anyway, so those are two filters that I have. So if you guys are interested and you want to see like, hey, like let's switch to bright field or let's switch to that blue filter, we can definitely do that. So that's what this filter is right here. I've got the blue filter, and then I can adjust the exposure as well. Well, you can't really see that. Sorry. This is kind of like that James Bond thing from GoldenEye, you know, the little circle. You can adjust that, and that adjusts the amount of light that comes in. And just another quick example of that, you know, so here's, sorry, my thumb was in the way. So here's all the light, and then here's less of that light. So you get a little bit more detail. Kind of cool. Now you guys are probably saying, come on, Jordan, let's get to that live stuff. We'll get there in just one second. I wanted to show you guys just the dark field, and then two prepared organisms, and then we'll switch over to the live. So this is dark field. So this is, the way that this works is that there is a black circle that blocks light from going straight in, and then the only thing that can illuminate the specimen or the organism is light coming from the sides. So we see dark field a lot. Dark field is my favorite thing to film with, and the very first thing that I saw with dark field, and again, so my dark field works really well at the two lower magnifications, not so much in the highest magnification. So I'll just kind of show you guys. It's just because of that focal distance. I think that's the word. Anyway, let me show you the thing that got me excited and wanting to buy a microscope. It was something very similar to this. This is called a Cyclops, and this one is stained, so it wouldn't look like this in real life. In real life, it's more transparent, and this is actually a female Cyclops. The way we know that it's a female is that the top little thing sticking out, that is an egg sac. And so this is my first time looking at this wet mount, but I just figured it'd be a good one to look at together. But yeah, Cyclops are really interesting. They are all over the place. They're found in lots of freshwater places. They're a microscopic crustacean, so they've got eyes and they've got lots of other stuff. Ooh, you guys are just full of great ideas over here. I'm gonna have to go through the chat at the end. I'm just seeing one about exposed negative film or something like that. That's a really good idea. Anyway, so here is that Cyclops. Just seeing something like this, and especially seeing it in dark field, was just mind-blowing to me. Let me see if I can adjust. Ooh, yeah, okay. I'm toying around with a new app as well that takes the camera stuff. So yeah, dark field, super cool. Cyclops, super cool. I think I've done a whole episode on them, maybe two. Let's back up. Another one, so okay, this is going to be the last prepared slide that I do, and then we'll go into the live stuff, because you guys have been super engaging and really awesome. Thank you all again for being here. Yeah, we'll talk more when I transition. So right here, if you can see, where's that camera? There's this tiny little speck. I think holding this up made it easier. So this is called planaria. It's a flatworm, and I also just made a whole video about these. It's planaria and snails that I saw under the microscope, and I actually saw them by collecting microbes at my sister's wedding a couple of months ago, and it was just, you know, I live in Utah and there's not a lot of water exactly where I live, and so when we're going to a wedding and there's like a waterfall, you know, at the venue, like, you know, I gotta go collect me some microbes. So if any of you other microbe hunters are out there like that, let me know. Maybe I'm just the only crazy one. Okay, let's get this ugly man out of the way and show you guys this planaria. So the interesting thing about wet mounts and prepared slides is that in some ways it preserves some detail, but then in other ways there are certain things that are missed. And these are probably created by a lab, you know, by technicians, but they might be using special equipment and stuff. And anyway, so this is actually a cross section of a planaria. So we're actually looking at it sideways. So if I was like this, like looking through its head, and so the interesting thing about planarians, so they're flatworms, and they don't have really a central tube. So you and me and worms and every animal that you're probably familiar with, the big thing that we share in common is that we all have a big tube. You know, we're all basically donuts. You know, we've got our mouth, and then we've got our rear end. Food goes in, waste goes out. But with planarians, they don't function like that. These guys are older than stomachs and intestines and things like that. So the way that they eat, they have like a little tube at the bottom third of their body, and that takes in food, and then it just gets distributed across their body. Now there is a circle in here, and you're probably wondering what that middle circle is if it's not the mouth or stomach tube. And that is actually, I believe, where its nervous system is. It does have like a, I think it's called a notochord, N-O-T-O chord, that is basically like a very, very basic brain. So again, very, very cool stuff, or at least cool in my mind. But yeah, so let me just take a 2 seconds here, and we'll kind of get prepared for our next little adventure. I'll remove this, and yeah, let's just check in with the chat for a second, see how you guys are doing, and then we'll jump into the live organisms. I've got 2 samples here, and these are both fresh, brand new. We got them today. It was raining, and but I just had to get something for this episode, something new, and it was fun. We were running around with my son, and yeah, got soaking wet and got some microbes. I'll make myself bigger for just a second. Let's check in with this chat. Let's see, a couple things back. The red egg looking things at the top right. Yeah, so back with that Cyclops, those were eggs of the Cyclops. So that was a female Cyclops. Usually they have 2. So they have just kind of one pair of eggs on each side. So that one was probably damaged or destroyed before it died, or in the process of mounting it. Let's see, make a Winogradsky column sometimes. Yeah, I haven't really looked at sulfur bacteria too much. And in fact, I haven't looked too much at bacteria in general. Oh, that reminds me. I gotta tell you guys. Okay, last thing before we go into the other live stuff where we're just gonna explore together. Let me switch over to PowerPoint again real quick and tell you, I've got some new things coming up. I wanted to give you guys sneak peeks at kind of some upcoming videos. So these are some things that are coming in the next couple of weeks. So I've got Great Salt Lake, as we've seen. You guys got a preview if you came to this thing early. And then I've got some lawn soil versus compost, and I've got both of those. I have some vials that are just sitting in water that I'm just kind of waiting for things to come alive in there. We'll do kind of the same thing with autumn leaves. Like we'll put some in some water and then see, what grows on them. Presumably there's going to be lots of bacteria and mold and protists, because otherwise, we wouldn't, we'd be filled to the brim with leaves. Microbes help break these things down. Big things that I want to work on is a food playlist, a microplastic playlist, and how-to videos. So you might have just seen I did how to look at microbes in grass. I'm going to do another one, how to look at, pond water, and just how to do basic stuff to get people into microscopy. Another thing, I've done a couple of food episodes, like we did honey under the microscope, where we looked at honey and agave. But this big thing that I'm missing is I can show you guys again real quick. So here's I can show you my setup here. Let's get out of manual and unzoom. So this is my microscope that I've got here. I've got my microscope holder, and then just a book that I've got, and then here's all my stuff. But one of the things that you'll see I am missing is one objective. So I'm waiting for my next month's Patreon donations. Those are going to pay for this next objective. And this is an oil immersion objective. I had one previously that came with this microscope, but unfortunately I left some oil on there and then it got destroyed. Some of the oil seeped in, and it was impossible to clean off. I've tried multiple times. So anyway, that's kind of prevented me from making this food playlist. I really want to show yogurt under the microscope and show bacteria that's used with food making, because there's tons of different foods that use bacteria, and then other superfoods like spirulina and things like that. Same thing with microplastics. Microplastics and nanoplastics can get really small, but they can show up in food. And so I've got some filters for it to filter out microplastics. But again, I'm just waiting to get that next thing. So big thank you to my patrons who are already patrons. Without you guys, I wouldn't have been able to get my latest filters that I've got. So that blue filter was one of 20 different ones that I got out of a pack. They're all different colors and they do different things. I'm going to do a whole episode on them. But anyway, so if anyone's got things that they want to do with really small microbes like bacteria, that's kind of why I've been waiting for those. So yeah, so let's go back to do this thing. Let's see. There's a couple other comments. Ooh, Massachusetts, great microbial mat. Yeah, magnetic bacteria. Okay, I've just heard about these. Do any of you guys watch Journey to the Microcosmos? I'm assuming at least one of you that here does. If you don't, those guys are. Somebody else is on my YouTube account. My name's Jordan Woods, and that's probably my wife. Anyway, so yeah, so Journey to the Microcosmos, they just did a really cool episode the other day where they talked about magnetic bacteria that are really cool. I've yet to see the full episode, but so don't spoil it for me. But yeah, I guess we can go to Massachusetts. Yes, so Matt Powers is doing research. Yeah, I'm always looking for collaborations. So if anybody is watching this and wants to do a collab, I'm all ears. It's just a matter of scheduling and fitting it in and remembering. I try to do one episode on my own a week, and then now I'm going to be adding these live streams if this works out. So Yeah, so who knows? Anyway, that's enough of that. I think it's time to look at some live stuff. So I have here two. Oh yeah, and let's get our sweet jams going back again. This is just like some random copyright-free lo-fi playlist that I found. So if you guys like it, let me know and I'll keep using it. If not, then I'll find something else. Yeah. So I'm Utah-based. Do you mean Nate, as in Nate from the internet? As in like the, what's their names? The King of Random. I sent an email out to the King of Random people a couple of weeks ago because they did some Microscope episodes and I was going to do something with them, but they yeah, I will. So I have an email on my About Me page. Sorry, I'm just trying to answer some chats. So I've got an email on my About Me page. I think it's just fantastic.microbe@gmail.com. If you want to shoot me an email, and then I can give you my cell and other information from that too. I'll typically just want to get an email before I know who I'm texting. Anyway, so I was at Utah Lake, Jordan River, right where the Jordan River enters or exits the Utah Lake, because it flows up, and it used to flow up all the way to the Great Salt Lake, and I captured these two samples. That now there's a dam separating the two, kind of like there was a bridge separating the Great Salt Lake that we looked at earlier. Now, so there's like a 50-foot dam, and so I collected some stuff, some algae on one side, and then some other algae on another side. So yeah, let's just go ahead and take a look at some of these. Now, eventually, with my next live stream, I have another camera that's just at my in-laws' house that's going to come in, or that I'm just going to pick up in next week. So I will have like a camera that shows like my microscope setup, so you guys can see me adding these things to the, what's it called? Putting things on the slides and getting them ready. So yeah, you guys are just only going to see half of that. Let me just try to collect some algae here. And that didn't work as well as I expected. I've got a couple of collection devices here. So I've got like some tweezers that I like to use, and then I have like some scissors. And usually I'll use like a combination of the two, or I'll just use my fingers and then just wash my hands after. Don't tell anybody. Probably the best thing to do, I mean, like if you've ever swam in a lake, things get kind of gross. But this particular lake is in the middle of an algae bloom right this second, right where I was, actually. So this is definitely water that you shouldn't drink, and it can be toxic, and and the main thing is, so it's called harmful algal blooms. It's basically when cyanobacteria and other like unicellular algae just kind of explode in population, and the bad thing about that is the chemicals that they release. So, okay, finally, guys, we're 40 minutes into this dang stream, and I finally have some live stuff for us to look at. You're probably like, oh man, Jordan, you should have started with this. Well, I had some live stuff at the beginning, if you guys were watching. Oh, yeah. Sorry, I was just kind of in the middle of setting that up. So, but yeah, thanks for the reminder. Okay, so let's get this filter on and just kind of see what we can see. Again, this is this is my first time as well as your first time looking at this. So, ooh, here it looks like we got something cool. This is, oh, here, I gotta switch from autofocus to manual, otherwise this thing is going to literally freak out at me. Do I just, oh, come on, dude, it was working 2 seconds ago. Okay, so we're gonna deal with this focus issue. No first live stream is without its issues, right? So, okay, this guy is definitely probably used to some more wiggle room. So, sorry, I'm just doing a couple more adjustments here, and then we should be able to see pretty well. Dude, I thought you could just... Dude, okay. Let's go in. Again, brand new app here. Because the app that I usually film with, it's called Pro Movie, or Pro Movie Plus, and it allows you to focus differently with the like you can separate the focus from the exposure and from everything else. So this is an insect larva. So we've looked at these things before, and it freaked me out the first time I saw one. I thought it was really interesting. They look like worms with like a special shell-like head. And they have tiny little feet at the top, and then they also have tiny little feet on the bottom. So let me know if you guys are seeing this. This guy is very, very active. It was just raining, you know. I don't know if that contributes to how active these guys are. But he seems to be... Oh man, see this autofocus, it's killing me. I'm gonna have to keep an eye on it. There, did I fix it? Maybe I fixed it. Okay, yay, good. Yeah, this guy is feasting. He's having a good old time. Let me just, I know some of you on the stream might be a little bit squeamish with insects and bugs, but man, the stupid focus thing, I swear. I'll just jump in. Yeah, so this is as close as we can get. It's really hard to see because there's a big clump of algae right here. But that is what they look like. Let's jump back and look. Yes, he does look very, very hungry. Let me jump back and switch over to dark field, see if we can get a clearer image of this dude. Hello, where'd you go? Oh, there we go. Yeah, so the interesting thing about microorganisms is that so many of them are like totally transparent. And so when they're on top of other things, it's kind of tricky to get a good view of them. Let's see. Sorry, I'm trying out one more thing with the camera. Yeah, cool. I'm gonna save this camera configuration, and then that way you don't touch it anymore. Okay. You guys, this is gonna kill me if I keep focusing on this, and I know you want to see more stuff, so we're going to keep doing that. So, yes, very interesting. Oh wow, oh awesome. But we've got plenty more microbes to see. So, there was one little guy that was moving around here. Without the ability to adjust the exposure. Let's see. That's the wrong one. This one. Just testing out. Damn, not that. Aha! I think I finally locked the focus. Okay. Oh, jeez, flippin' wheeze. Okay. So, can I also adjust the white balance and get rid of... Oh no, that's not the one. It's the exposure that I need to find. Okay. You guys, I'm really sorry about everything that I've done. I've totally just destroyed this whole thing. Okay, set everything back to auto. I figured out how to do the focus here. I just adjust it like that. Cool. Let's get out of dark field because when I get out of dark field, it's a little bit easier to see other organisms and stuff. So, okay, so there's our little larva. It could be like a midge fly. That's frequently what they look like. Let's see. Let me know if you guys like. So I've got two options with my bright field. I've got the blue background right here, and it kind of makes the background a little bit blurry as well. Or I have the this crisper view, which has like a wider background, but you can kind of see a lot more. Oh hey, look, we found another thing. It looks like we have another larva here. Let me see. If they're digesting what they eat, is this by some acid, or are there even smaller things that live in their microbiome which break things down even further? That is a good question. So I think with, so don't quote me on this 100%, but I'm pretty sure, like I know that insects have a microbiome, but blue 5 out of 10, sepia 7.5 out of 10. Yeah, there's I know they all have kind of their pros and cons, but anyway, yeah. So I know that insects have a microbiome. I don't know about protists and stuff. I'm assuming that they do. I mean, they do eat microbes, you know, and so I'm guessing that some of them still just hang out. Okay, let's adjust our focus, see what else is going on here. So if I zoom here, now we're kind of looking at the floor of the glass slide. So it looks like there's a tiny microorganism right here. I could zoom. We just like seeing it, Jordan. Well, thanks. I like you guys seeing it as well. Thank you guys for showing up. Ooh, this one's cool. Okay, so this right here is called a rotifer. And this, again, is also one of the very first things that I saw in a microbiology class. And so these are very, very cool. These are multicellular, so they are animals, as opposed to the single-celled things like the algae that we're looking at. Other things that are single-celled are like paramecium. You'll see those. And amoebas are single-celled, stentors, gastrotrix. No, gastrotrix are not. They're multi-celled. But yeah, so this rotifer, it's got one leg and a couple of toes. Dude, I was just talking about you, man. Where the heck did you go? Well, it got nervous. It got camera shy. So the cool thing about them is that these guys feed by spinning a little crown of hair around their or they don't like spin the hairs, but like they beat the hairs kind of like in a spinning fashion. You can see that one was just using it to travel. They use it to travel, but also like once they anchor their foot down, they can use it to feed. Like it creates like a vortex, like swirling bacteria towards them. So yes, yes, MF microbes, yes. So yeah, we've got some rotifers here, and you can see one of them looks like it's just kind of sleeping or hanging out in the middle, and then this other one on the left is pretty lively. I haven't gotten this question on the stream, but yes, it does have toes. It has just a couple of them, and there are different species, and so some have like 3 toes, or some have, you know, 2 or 1, all just to help them grab onto things. So this is right at the edge of the slide, so it's a little tricky. And this one is also, I believe this one is a female, because it has a very large, dark ball inside of it, little globe. That is definitely, I would venture to say that it is a seed of, or an egg of the future baby. Couple cool things about rotifers. These things have been, they have a remarkable ability to freeze themselves, or go into hibernation and come out of hibernation a long time later. So how long, you might ask? Well, they were just found in some ice recently, I think in like Greenland or Antarctica or something. They found some rotifers that had been frozen for like 50,000 years. And they were able to reanimate just by melting the ice. So they basically they shrink down into these cysts, like they remove all of the water from their bodies. Lots of other small organisms can do this too. Like this organism can do this as well. So this is a ciliate that we're looking at. And so this is a single-celled organism. So this was the same size, more or less, as that rotifer. And sorry, I hope one of the things that you guys notice is, so I don't know if you're longtime viewers of this channel or if you're not, but I try to get like the most crisp, clean footage possible with these, and so I often spend a lot of time just chasing these guys down, and it is really hard to follow these things across the slide, because like I've got one hand adjusting the focus, and then the other hand like I've got this finger, like these 2 fingers going north and south, and like these 2 going like sideways, and so like I'm chasing these, sorry, I'm switching to the chat real quick. Do rotifers have DNA? Yes, rotifers do have DNA. And yeah, so just like any other animals, and their genomes have been sequenced like a lot of their species has. And so that's kind of cool. One interesting thing about rotifers is that there are several species that are only female. So they are able to produce eggs that just self-fertilize, and they've been living without males for tens of thousands, if not millions of years. So there's lots of microbial species that can do that, that live without females. I don't know of any species that that they live without the males. I don't know of any species that lives with just males and without females. Sorry, the female-only ones are the ones that exist. Yeah, someone says, what are we looking at? So we're looking at a couple samples from Utah Lake. Earlier in the stream, if you go back, this whole thing will be posted later. We were looking at some stuff from the Great Salt Lake, talking about microbes that make the lake look 2 different colors. Like the entire north half of the lake is pink, and the other half is kind of bluish green. So, okay, let's take a look at some of these things here. So to the untrained eye, it might look like we're just looking at a bunch of scum. But if you just sit and wait for a second, geez, my wife needs to just calm the heck down. Or get her own channel so she can comment as herself at least. What we're looking at right here is we have a couple of, I mean, there's a lot going on, but we have some diatoms, and those are the things that are moving. Let me zoom in a little bit to give you a clearer picture. So this is our highest magnification that we've got currently, and these really small, so this big long pipe looking thing, that is a diatom. So it's a unicellular algae. And diatoms come in all different shapes. So they're like circles, they can be like star-shaped. A lot of them look like, what's the word? I think the word is pennate, P-E-N-N-A-T-E. So just, yeah, Google diatom shapes and you'll be amazed. And like they're just like so many cool things, and like some of them look really cool 3D. But yeah, all of these really little, not the tiny little squiggly ones, like those really, really small dots, like those are all bacteria. But those other sticks, let me maybe zoom out and see. Oh yeah. So here's a couple other ones. So these are all different species of diatoms. So there's one at, there's one here. I'll move my face for just a second. And so that one's moving. And these two right here. Now diatoms are really interesting. And oh yeah, here's a rotifer. We've been looking at these. So here's one close up. These also have eye spots, and so they're able to sense light and dark, and they also can respond to certain stimulus and stuff. So there is a basic level of consciousness and intelligence that one could argue with those. Anyway, so back with these diatoms, the cool thing about them is that they have a really strong outer shell, which is based out of silica. And so I've seen like paramecium and other ciliates try to eat these things, and their bodies just cannot digest them. And in fact, it's because of this that diatoms actually make really good fossils. And so if there are any farmers out there, you guys might have heard of diatomaceous earth, or any gardeners. Diatomaceous earth is literally just dried up fossilized diatoms. They could be fossilized as of 2 days ago, dead, and or from millions of years ago. But yeah, so that's kind of what we're seeing. There's lots of diatoms here. These big circles, these are air bubbles. Oh yes, Speak Life, Garden, Homestead, and Permaculture. Now that title sounds like you probably know what I'm talking about. With diatomaceous earth. That's actually something that I haven't looked at under the microscope. Yeah, maybe I'll look at that and then compare that, like diatomaceous earth, with some live diatoms and see if I get different shapes and stuff like that. That'd be kind of interesting. Let me back out and let's see. You guys, I've got a confession to make. And the confession is, I really want to keep this live stream going and keep showing really cool stuff. Oh yeah, so this is all algae. So it's diatoms are like the unicellular algae. Now these are algae that have distinct cells, and you can see the difference with the cell walls. Yeah, dude, we've got so much stuff going on here today. What a great time. Hey, thanks, Nader Anderson. A+ work. Well, I do it all for you guys. Oh, while while we're just hanging out here looking at this larva, I wanted to check on something. I told you guys that with this stream we were going to be celebrating 4,000 subscribers. Well, I have to say we are almost at 4,000 subscribers. The actual number that we are at is 3,995. So if you are watching this live stream and you have not subscribed yet, you could very well be the 4,000th subscriber. And that could be very interesting, and I would very much appreciate it. There are all sorts of benefits to being a subscriber, like you get to hang out with me whenever I post some stuff. Every time I post a video, I'm always there like for the next hour or two answering questions. I don't know what this thing is in the middle here, but it looks very, very interesting. This could be a what's called a testate amoeba, which is an amoeba that finds things to make like a shell around it, but I wouldn't be able to tell without filming it for a little bit longer. So here's what I'm going to do, you guys. I have a whole other sample. That was just... Here, let's get Big Jordan on for a second. Guys, that was just one tiny little drop of water from this. So there are plenty more of things to look at with just this sample. Now, with this channel, I try to travel all over the place to get really cool footage. But honestly, I could probably go to the same place every single day and find different things all the time. And in fact, that's what I do. I usually go to Utah Lake and Jordan River, and that's kind of my main thing. I also have just like a bucket of water outside in my backyard that just collects rainwater and whatever other gunk that the wind and dirt bring in. And I empty it out probably like every 3 months, but it's always a new surprise. We've got sample number 2. I'm going to keep going probably until about 9:30, or unless you guys, I mean, you guys are always welcome to go if you want, but yeah, so I might stay around for a little bit longer. I'm feeling awake, but let's see. Let me just look at some questions real quick. Yeah, there's a reason, Jordan, on the male thing. Yeah, women are awesome. So, at least in this species, there are some women that get females that get pretty ferocious in other species, but also some males that get pretty ferocious in other species. Yeah, go ahead and send your diatomaceous earth over here. Even just like, you know, something as small as this, like get a little Ziploc bag, I'll shoot you my address, you can send it over. Yeah, crazy amount of life all around us. Mucus under the microscope. Yes, we did look at our nose under the microscope. It was during the months of COVID. That was kind of crazy. Well, I haven't done just plain mucus by itself. Like, we've grown bacteria from our nose, but I haven't, like, you know, taken a long strip to look at. Yeah, there's a lot of weird body things that I'm like, ooh, I should do this, but maybe it's too gross. Like mucus, or some other microscope YouTube channel just did fecal matter, like literally poop under the microscope. Yes, my email, I'll put it in the chat here. Microbe@gmail. Yeah, that's my email, something like that. Yeah, another one that I was thinking of doing, and maybe this, I don't know if YouTube will like this, but there are some videos out there of sperm under the microscope, but I wanted to see like sperm versus spermicide under the microscope. Again, a little risque, I guess. I don't know if I'd have comments on that one, but I don't know, like, people want to see stuff, and so I don't know what people want to see. Do they want to see like the gross human body stuff? Do they want to see gross food stuff? So I'm just preparing this next slide, by the way. Yeah, let me know what you guys think. Yeah, so about sending samples, I'm still a little iffy about taking samples from people if it's from out of state. Just because I've looked up laws, and like there are certain laws that like you can't transfer nematodes and other things like from state to state. I was actually going to collect microbes from Hawaii, but when you get on the plane there and back, you actually have to declare you know, anything living that you're bringing back. So that's, but maybe there's other options, and I just don't know about. But at least in the United States, that's how it is. So okay, new sample here. So this algae stuff looks a little bit different. So I've got a little bit of dirt on the bottom, and then there was just like some green sludge on top of this dirt here. Yeah, people sell nematodes for gardening. Yeah, I don't know if it's like about the species and if it's just like across state lines, or if it's across like I mean it's all unregulated. And the other thing too is that like the amount that I would be collecting is so small, like a little tube like this, and then you know I'll bleach it or flush it down the toilet. And like the only thing that, you know, if you do have microbes that you collect, like they recommend, you know, putting it back where it came from, or you can flush it, because that's a good way to, like usually they're in such small quantity and there are so many microbes in the sewage system. But okay, sorry guys, I'm just trying to like answer your chats and do all this at the same time. I just gotta focus on one thing. I don't know if you can tell, but I a little bit about me. I have what is called attention deficit disorder, and it is very, very strong, especially when I'm doing microscope stuff. And so this is quite a challenge to do the chat and all of this stuff. But you guys have been really great sports. I really appreciate you guys all watching this. Yes, do not mail us your poop. I will always accept non-living things. Like if you want to send, you know, like that diatomaceous earth, like I think that's a great idea. Or like if you have like butterfly wings or bird feathers or anything that you think would be cool under the microscope, that's not alive. Great ideas there. Yes, but yeah, I will not accept poop or fertilizer that comes from any animal. So yeah, let's see here. Are you guys liking the stream so far? Like, do you like this pace? Do you like this style? I'm like totally open. Again, like this is the very first time I've done this stream, so like if you're like, hey, more microbes, less stuff, like, or if I'm not focusing on certain things for long enough, let me know. Yeah, let me switch over to dark field for a second. Yeah, I think, you know, maybe just kind of looking at this a couple of times, like we'll kind of see, or like doing a couple of live streams, it'll give us like an idea of what we like and what we don't like. Okay, the sun. There's a picture of the sun on my thing here. There we go. Okay, I'm figuring this out. Okay, so I think we've got lots more of those diatoms here. This is kind of what they look like in the dark field. But these could also be long strands of bacteria as well. Yeah. Dude, focus. Where the heck did you go? I thought I had you on lock. Okay. Okay, so some of these for sure are diatoms, but some of them might not be. Overhead projector microscope for classroom use. What? That sounds really cool. James, do you have like is there like a name brand of that? Like I've never heard of those before. If you know the name of it, go ahead and drop it in the chat. Yeah, so these diatoms are... Well, a really cool thing to do is if you're ever filming diatoms, you can like make a time lapse of them, and it's nice. You can see them move a lot more easily. Whoa, hold on. Gotta back out of my dark field real quick and adjust the light. So these long strands here, besides the... Oh, hold on. I got a weird thing just pop up. These look like they are this thing called oscillatoria. Oh, you know what? Maybe they're not. That's the thing with live streams, is that I'm just kind of like spitballing with things. But these things could be a type of cyanobacteria called oscillatoria, but I'm not sure. But then, yeah, right next to all of these other diatoms. Yeah, so this is kind of like where you'll see diatoms. So, as if you're just tuning in from 2 seconds ago, this is what this came from. Sorry, it's kind of tough to see with the lighting here. But it's really just dirt, or like wet mud, just like right under the river, and it just had like this little thin layer of gunk on it. And that gunk is these diatoms. So, Kenavision X1000. Now that sounds like a tool, like a microscope tool. I always like how companies just add the word, or like they add like crazy numbers at the end. Does that make this better? I feel like I'm an optometrist. Do you like 1? Or do you like 2? It's over 9,000! Thanks. Thanks, cute wife, for the over 9,000 meme. Yeah, fellas, get yourself a wife that does Dragon Ball Z memes. Yeah, also, as far as like live streams go, like, again, ooh, guys, we found an amoeba. Oh man, this is, I'm telling you, the end of summer, like, this is the golden hour of microscopy, because everything is alive, everything's blooming, everything's having a good time, and, or maybe not everything's having a good time, but there's a lot of things in high population numbers, and so you're a lot more likely to find things. It's kind of tricky to find stuff in winter, so I'll probably be collecting a bunch of microbes and storing them in my basement to look at. But yeah, so right in the middle here, this is, right where my face is, right there, is an amoeba. And this is the highest magnification that I can get in. And it basically is very, very clear, and it moves very, very slowly. So most of the amoeba footage that you see online is sped up. I usually film these guys on time-lapse videos and stuff. And yeah, like I said, there are different kinds of amoebas. Some of them enclose themselves in shells, but the ones that most people are probably familiar with are these nude ones. I don't know what the official name is, but there's hundreds, probably thousands of species of these things. So, but yeah, anyway, they have like little, so it's just one body, one single cell, but they have like special proteins that they can use. Change background to show amoeba. Yeah, maybe I can make this a little bit bigger. Yeah, here. Yeah, there we go. Okay. Because this guy's not moving anywhere. Okay, let me see just how big I can make this. Oh no, it's gonna get run over. Okay, well, he's probably okay. So, by the way, I'm doing this, yeah, what the heck, stick guy? Which was the diatom right there, with a couple of other diatom hitchhikers, it looks like. So, for those interested on my camera, so this is the iPhone XR. It's capable of 4K, but when I'm live streaming, I think it jumps down to 1080p, or maybe even 720. And then the other thing with this phone is that it does digital zoom instead of optical zoom. So zooming in on this, like, we are losing loads and loads of data. My mom has the latest like iPhone 13, and so I'm gonna bring my microscope over to her and see how it compares. I'm like, yeah, I've had the same phone for years. I've been in the market for a new one. So yeah, maybe it's time to upgrade to the 13 or the 14, because both of those have optical zoom, which means that you don't lose data when you zoom in. With digital zoom, it just kind of fills in the gaps for you. One cool thing about amoebas is that they have several different behaviors, and depending on what they're doing. So if they're hungry, like, they will like stretch out their limbs in one way or another, or if they're about to divide, then they'll, move their limbs in another way. Oh shoot, just bumped it out of the way. Bye, amoeba. Oh, you guys. Oh, did I just lose it? That's embarrassing. Amoeba, where did you go? Guys, I lost our amoeba. Oh, no, it's right here in the middle. But yeah, so we are like crazy zoomed in right now. So that's why we're losing a bunch of information. But there are certain amoebas that can get a lot bigger, and those ones are sold by biology teaching companies. Yeah, he gone. So anyway, let me just zoom on back. I don't think we're gonna see that guy again, but that's fine. Anyway, as I was saying, so they have different behaviors. Like, they'll go in attack mode, and then they'll go in like sniffing around mode. And with each of those different modes, their feet kind of move differently. Sometimes they look like blobby, and sometimes they look, spiky. And yeah, I'll do a whole episode on that eventually. But yeah, so this one is, we're just seeing lots of diatoms. It's been really great. Yeah, really appreciate you guys that have been sticking around in this. I think this has been a lot of fun. And I feel like the more, that I'll do live streams, the more I'll get to know you guys, and the more you'll get to know me. And we'll have a lot of fun with this. Again, yeah, I was just kind of planning on doing this till about 9:30, so I'll probably wrap it up in just a little bit. Because I do have work in the morning. I am a real worker. What do I do for work? Oh, nobody asked? Oh, well, that's okay. Just kidding. So, I don't know what this thing is right here. If this is another diatom or if it's something else. No, because it's got cells. I don't know. I'll have to look back in my book, in my guidebook, to see what that is. Yeah, that one's interesting. What do I do for work? Thanks, Matt D. I work for NBC Universal, so the, Jurassic World, Minions, those guys. I'm actually, so I just started with them this year, as like a contract to hire job. So if they like me, then they'll hire me, presumably in like a month or two. And yeah, so I'm a business analyst, and I work on their behind-the-scenes websites, which are all built on this software called Salesforce. So some of it is like sales related, but like there's also lots of other custom websites as well. And so yeah, so I'm the business analyst, like the in-between guy. So I talk with the tech people, and I talk with the business teams. And yeah, so not very science related at all. I didn't do too well with tests back in the day in college, and couldn't make it through like organic chemistry and stuff like that. Otherwise, I probably would have stayed through and become a pharmacist, because I was a pharmacy tech for a while, and that was my career plan. But yeah, you know, life changes, but I bought this microscope when I was, you know, doing pre-med, pre-pharmacy. Yeah, lots of little unicellular algae right here. I'll have to go back in and ID these things. Yeah, very, very cool. I'm a professional nerd. Yeah, thanks, wife. Oh yeah, hey look guys, we have another amoeba. This is at the same magnification, so this one is a lot bigger. And then I could probably do that zoom thing again. It might get all blurry and gross, but this is what we've got. Yeah, so usually they kind of move around with just one big foot, and then just like move everything else around it. Let's see, can I adjust anything else? Make this a little bit better. This is full. Sorry, I'm just doing stupid things with this right now. Let me just fix that. Okay. So, yeah, a lot of you guys are probably like, why doesn't this look like the stuff in textbooks? Well, there are better microscopes out there, better compound microscopes, and then the way that we get really good views is through like electron microscopy, where they literally fire electrons at things. Like any science magazine that you've seen at the doctor's office probably uses electron microscopy for their photos. Can you grow amoebas and sell them? Me as a person, or just like in general? Like amoeba cage fighting, you know, that's actually pretty interesting. So I'm reading a book right now. It's called, oh, it's not in here, but it's about the consciousness of microbes, and it's essentially this guy's thesis where he talked about, he gave all of these examples of how different organisms respond to different stimuli to basically prove that microbes have at least some level of consciousness. And one of the examples that he brought up was amoebas. And he said that, you know, studies have shown that amoebas are capable of recognizing their own kind, their own species, and there are certain species that will eat themselves, like they self-cannibalize, like among their species, like they'll eat their sibling or whatever. But then there are other species that only eat other amoebas, and then there are some amoebas that don't eat either. Like they don't eat themselves, or they don't eat other amoebas, they just eat diatoms and bacteria and other stuff like that. Yes, there we go. Minimal Selfhood and the Origins of Consciousness by R.D.V. Glasgow. It's a little bit of a heavy read. It's literally some guy's thesis. And, but yes, that goes back to my professional nerd thing. I left the science major, but science I never left at heart. Like, if you look back in my library, like I have tons of books on animal consciousness, tons of books on biology, invertebrates, microbes, big animals stuff, books on space, and I've got some Stephen Hawking back there, some other, well, I haven't read that one. I'm not going to say that I've that I've got these big name books and that I don't even read them. Do you guys ever do that? I think that's another big ADHD thing. Like, I get big ambitions to read like these huge books. Like, I'll go to Barnes and Noble and buy like these just ginormous books, and I've read a lot of them. One of my favorites is it's called Comparative Psychology, and it's all about, yes, I do have basically a whole shelf on microbe consciousness. But can you see the mitochondria on the amoeba? I don't think I could with this resolution. Maybe there are certain amoebas where I think it would be easier. You can also stain amoeba, or like you can also stain the organism, so you can see them like certain stains adhere to certain parts of the amoeba and like of other cells, so like they would adhere to the mitochondria, so they light up. I haven't done that specifically, so, but yeah, with this particular one, it's too grainy, you wouldn't be able to see it. Most of the ones that I've seen are too small. Yes, I've also got Calvin and Hobbes and some other stuff. I'm a pretty fun guy. So let's zoom out. Let's just take another little glance around, because I'm having a little bit of fun here still, and yeah. So again, different shapes. Oh, here's a unique shape that we haven't seen or talked about yet. 2 weeks goes by and it's 0%. Oh, every book I've bought with the intention to read, that's the overall. Yeah, pretty much. I gotta show you guys these huge books. So this, we're looking at some diatoms, by the way, for those who are tuning in and that don't want to listen to me. But anyway, so this is back when I was in college a couple years ago, and I just wanted to be the most impressive person in the world. And so when I was at, look at these things. When I was at Barnes and Noble, I just walked around for like an hour, and I was like, where's the biggest book here about like science or knowledge? And I found this. This is Gray's Anatomy. It's not the TV show, like it's literally this book by Henry Gray, who, like, it's just an anatomy textbook, mostly text and a couple of pictures. I thought it would help me pass my anatomy class. One of the very few classes that I failed during college. Not because of the book, but because I got married halfway through the semester and just was not, I think I failed all my classes that semester. Oh, hey, hold on. Pause. We found a cool little microbe here. Ooh, very cool. So this looks like another ciliate of some sort. So when you see me on my channel, I mention the word ciliate a lot, and that is because there are a lot of them. There are probably like, whoa. Sorry about that exposure brightness. A ciliate is basically any single-celled organism that they kind of look like paramecium. They're covered in little hairs. Some of them have specialized hairs at the end, and then some of them have specialized hairs at the front to help them swim. Some have specialized hairs on their feet, which help them walk, but most of them kind of swim around. So that is what that is. Anyway, so yes, I bought that thick book because I thought I would impress my wife, and, well, she was just my girlfriend at the time, and just other people in my dorm. And then the other one is this right here. This is called On the Shoulders of Giants by Stephen Hawking. I've taken very few physics classes, but it's basically, he goes by all of the writings of these physicists and explains how math works. And for at least 2 seconds in a bookstore, I thought that this would be a book worth reading. And so it's sat on the shelf for about 6 years now. But it's kind of cool. It goes through Copernicus, Galileo, Da Vinci, Isaac Newton, and Albert Einstein. You can see the bros right there. Never gonna read that. But it makes me look smart. So there you go. Yeah, you guys, this was a lot of fun. So again, just to kind of wrap up, like this will kind of be the, ooh, you guys, there's lots of bacteria here. Cool. So this will kind of be like how it goes with my live streams. So sometimes I'll grab some new stuff, but other times I will, the bros. What is this thing, you guys? This cool worm-like thing has a really long tail at the end. I don't think I've seen something like that before. Dang it. See, this is why this is such a bad hobby, is because you can never look away. Or like, you can never stop. Okay, well, if this is going to be like this, then oh my gosh. Okay, now we're in Grunge Fest mode. Let's get out of all of that. Well, whatever that worm was, that was really cool. So I'm going to be holding on to these for a while. Anyway, so let me just back off of that, and we'll close up shop here for a second. So let me just wrap up my thing and give my big thanks to you guys. Let's see, I've got one more slide to make it look cool and professional. And let's see. Look, first live stream. I had this really cool PowerPoint, and it worked, and I had music, and I think that worked, and I had all sorts of fun stuff, and actual humans showed up, and some of them weren't my mom or my wife, so that's cool. Anyway, you guys, so again, this will kind of be the way that it's set up. So I will look at either some new stuff if I don't have anything, or like a lot of the time I come out with my new episodes on Saturday mornings. I call it my Small Adventure Saturday, and so like that's where I go places and like come up with the cool story, cool stuff. If I have any leftovers from that, we can take a look at those things as well. So if any of you guys like have questions or more things that you wanted me to look at from those, I'll hold on to those at least until these live streams, and then I'll toss them. I got in a bad habit of not tossing my microbes for a long time, and I had shelves of things, and like there's always cool stuff to keep looking at in them, but I need to start tossing them out more frequently. But anyway, I could probably do this for hours or years, which I thought I would only be able to do this for a couple of minutes, which was crazy. I guess if you find good pond water, the microbes will just show up. That's the other cool thing about doing this, is that every single time it'll be something different. That's probably why, like, there's only... Oh, when do I give out the $10? Did I say I was going to give out $10? I don't think so. No, when do I get the $10? Wait, you guys, last thing we got to check. Did I get my subscriber goal? We're at 3,998. We need 2 more subscribers. 2 more. If any of you guys are not subscribed, or if you have a second channel, or for some reason, anything else, like, please, we are almost at 4,000 subscribers. There's 2 more chances. You can be the 4,000th. You will not get $10. There's no reward for it other than being cool. I'll give you a shout out at the end, like of my next episode or something. But yeah, so here we go. It was really fun, really great. Yeah. Oh, I was going to compare Microscopy to the other live streams that I watch. The only other the only other live stream channels that I watch a couple of gamers. The only game that I watch is Super Mario Maker. Kind of a dork, but the reason that I love it is that there are millions of levels because everybody makes their own stuff. And this will come back to microbes in 2 seconds. But that's why I really like watching it, is because it's always something new, that's kind of people our age, I guess, or this generation's all addicted to novelty. But I think that's why I like microbes as well, because like every time you look under the microscope, it's going to be something new. So you can pretty much be guaranteed that it'll be a really fun time. We'll be learning together, and it'll be awesome. So thanks again. And just wanted to say, see you guys next week if I don't see you before then. And yeah, so again, every Tuesday at 8 o'clock, I think we've got enough people that have showed up to convince me that this is something that some people might want to enjoy. So yeah, next Tuesday, 8 o'clock, same thing, different microbes. All right, thank you all so much. See you later.